My name is Atul Butte. This is actually my first time to ISB, so thank you for the invitation. I'm actually ashamed. It's taken me forever to get here, but I'm glad I'm finally here. I'm a medical doctor, speaking to a couple medical folks here, so I'm just going to start with my conflicts of interest, just a few. Suffice it to say, I've started a bunch of companies. I consult for a bunch of companies. You don't have to believe anything I say over the next 30 minutes. I wouldn't blame you. I'm most proud of the bottom right, though. Those are all the companies started by my students. More than half my graduate students now start companies, even if they go into academia. Because in my lab, we do have a saying that if you want to change the world, you can't just keep writing papers about it. You have to file IP, and if no one licenses it, you got to start the company yourself, even if you go into academia, because academia is compatible with it. So a lot of places, it's taboo to talk about companies, not for me. So I'm going to mention companies a lot during this talk. So I'm particularly proud of that. And they start the companies, and I start my companies with the most amazing platform in the world, and that's data. It's often big data, and it's really often open big data. So I'm going to talk about how they do it, and I'm going to talk about how I do it. If you haven't heard, we're in the middle of this precision medicine revolution now. I know it's been said already a couple times so far today. And obviously, we're already 1 year into it. We just had a 1 year anniversary. A couple folks went to the White House, and all my colleagues were asking me, so what did you discover in precision medicine over the past year? And I've discovered there's a lot of money in this field, actually. Beyond that, I'm not sure what else we've discovered. But obviously, a lot of people writing grants for this, all sorts of kinds of grants. But actually, precision medicine makes physicians very nervous, if you haven't figured this out. It makes physicians very nervous because if we're moving to an era of precision medicine, by definition, the last thing we were in was imprecise medicine, right? By definition. And so how do you even tell? I mean, how do you convince physicians we were in imprecise medicine? Well, how do you even measure the precision of a medicine, right? Let's start with that. And so to me, I think about how we measure the precision of medicine based on the categories of diseases. How many kind of pigeonholes do we have to put patients into? How many diseases do we have? And so the concept of this goes back almost 200-300 years, this concept of classifying diseases. The word for this is called nosology. Nosology has nothing to do with your nose. It's a systematic classification of diseases. And so I buy books like this. I mean, so these are books, very old books of how we classify diseases. The current system we use is called the International Classification of Diseases, ICD. You know, we were on ICD-9, we were moving to ICD-10, I'll tell you about it. And I buy books like this. If you're curious, you can come up the front, ICD-2. This is second revision from 1909, so this is a 100-year-old book. You know, when old doctors die, they throw out this stuff, actually. So this is like trash you can buy on Amazon. And it's amazing because, I mean, the book from 100 years ago, it's amazing. There's a code for appendicitis. I'm just flipping through. There's a code for cancer of the larynx. But then literally, it says here, literally on page 141, if you see a patient that died because of a visitation of God, use code 189. Literally, this book has to say that, right? You want to know imprecise medicine? This is imprecise medicine, right? Now we're moving ahead, right? So we are moving ahead towards, this is ICD-2. We've had ICD-9 for about 30 years. And just last year, we moved to ICD-10, the 10th edition. And actually, the physicians, again, were revolting against this because there's so many different codes, right? And in fact, they started to make fun of the codes. Believe it or not, there's a code that if you're surfing and get hit by a whale, there's a code for that, actually. I mean, there's code. So people are making fun. My favorite meme that went around the internet was this one. I don't always get sucked into a jet engine, but when I do, I use code V97.33XD, okay? Now actually, when I give this talk in San Diego, okay, it's actually a naval base, right? And there are a lot of people that recognize people do work on the surface of aircraft carriers, and you can get sucked into a jet engine, actually, that is true. But this is the chronic form of the disease, okay? So I can imagine getting sucked in once, but not twice. How would you do this twice, right? But there's a code for that, right? So people are making fun of ICD-10, but actually what we should have been making fun of is ICD-9, okay? I mean, this is what we've had for 30 years, and this is my favorite way to poke fun of ICD-9. This is standard. This is still the code in our systems, right? Take lung cancer, #1 cancer killer. How do we code lung cancer in ICD-9? Here, it's 162.9. Do we take advantage of all the molecular testing we have, the sequencing of oncogenes, EGFR and stuff? No, of course not. Do we even take advantage of the microscope, right? Distinguishing squamous cell from large cell, from adenocarcinoma? No. Do you know how we coded lung cancer in ICD-9? left lung or right lung. Literally, that was the code for lung cancer. And this is what we should have been making fun of, and the ICD-10 doesn't even fix this. And what we're talking about precision medicine is way more than that, way more than just what even ICD-10 is. We have to come with better codes and better knowledge and better molecular definitions of our diseases so we can tell each other in medicine what our patients have and now tell the computers what we think a patient has so the computers can help us. And that's the era of precision medicine we need to get to. Of course, enabled by all the molecular testing I'm sure everyone's been talking about all day today. This is my favorite example to talk about these kinds of omic technologies. On the left here is an Affymetrix gene chip. I got my start about 17, 18 years ago looking at the first 50, 60 of these in collaboration with Todd Golub and others at the Whitehead. And these chips are amazing. The size of your thumbnail, actually this company Affymetrix kind of doesn't even exist anymore. They just got acquired, sadly. But I still love this chip. It looks kind of cool. In fact, it's so cheap now I can carry one in every suit, right? So if you've never seen one, this is what they look like. Big data comes from small packages, just like this. And actually, so everyone runs these, right? It's not even a big deal to run microarrays. In fact, we've moved past this. We have RNA-seq. Of course, we have other omic technologies too. But what's amazing with these is when we got tired of measuring them one by one, we started measuring them in the 96-well plate. And that's so cheap now I can carry that in every bag. And I have one in my hands here. In fact, when you get tired of measuring them in the 96-well plate, there's a 384-well plate. And that's so cheap now I can carry that in my bag. I cannot more clearly illustrate exponential growth in biomedical data than to show you these plates. And we don't even use these anymore because we've moved on to RNA-seq and other things. And that's how fast biomedical data is growing. The vast majority of researchers, they run one of these things, they get an amazing paper out, they deposit the data into the repositories because the journals make you do it, and then they move on to collecting more data. And the repositories, that's what really is the story here, just continue to grow and grow. So when I got my start, there was no such thing as a repository for this kind of data. In August of 2012, this is a paper came out in Nature, featured my lab and others. In August of 2012, we hit a million of these chips publicly available, 1 million. And actually, if you look carefully here, it looks kind of sad, right? Doesn't it look like it's slowing down here at the end? And then you realize they only counted half the year, right? This is August of 2012. Still doubling every 3 to 4 years. In fact, we're just a few thousand microarrays shy of 2 million publicly available samples. That means someone's colon cancer, someone's breast cancer already run on this or RNA-seq or anything similar to this, data completely freely available. What an amazing time it is. I know things are depressing in science and funding, but look at this, 0 to 1 million. And why do we do this? Because we have this culture in medicine and research that if enough researchers use this kind of data, you got to release it to the public. Okay, it started with GenBank, which is like 38 years old now, right? DNA sequence, first figure came out in a paper. No one wants to type that in again. You got to send it electronically, starting sending tapes to each other, right? And now if enough people use this technology, you got to release the data. Now, I know you can get lost in the billions and trillions and all of that, so I like to make it crystal clear. Any one of your researchers, any researcher in town here or in my town or my university can go to one of these two repositories, for example, NCBI Geo or Express, and let's say, I'll forget about our scientists, right? Any high school kid now who needs to do a science fair project, let's say she needs to do a science fair project on breast cancer. She can go to these websites, search Geo, for example, for breast cancer, 67,000 samples of breast cancer ready to download, about as easily as she can find a song on iTunes today, right? That data is completely freely available to everyone. I'm not showing you any magic that you couldn't do yourself if you have enough learning, okay? Everything I'm going to show you here is stuff you can do too in your own facility. I don't have any special microscopes or anything. I'm just showing you stuff that everyone can do here. Now, the number I like to look at is this one, 2691. Let's call that 2,700. 2,700 research groups and labs have already submitted data into this repository on breast cancer, okay? Let me tell you how ridiculous that number is. Let's pretend I'm writing a brand new NIH grant and I'm a complete newbie. I know nothing about breast cancer. And you're my study section. I propose to you, I'm going to get 2,700 of the best breast cancer labs in the world to study their most characterized patients with the latest technologies, and I propose they're all going to give me that data for free, right? If I propose that to you in a forward direction, in an NIH grant, you would laugh me out of the room, right? Why would 2,700 labs share that data with you, a complete newbie, for free? Yet here it is, right? Here it is. So I like to call public data like this retroactive crowdsourcing, okay? It's crowdsourcing because it's getting your friends on the internet to help you. 2,700 labs are there to help you, and they don't even know they're helping you. Actually, 2,700 labs are there to help you, and you don't even know they're helping you, right? I don't know, if 67,000 samples of breast cancer aren't enough, just wait another year, it'll be 100,000 samples, right? We're gonna add another couple hundred in this half hour right now, okay? Just doing nothing. That's how many people are just constantly submitting data. Wouldn't you want to use some of that data? And to start an experiment with the data instead of starting with a pipetter, right? That's the kind of concept here. And so our first kind of example, I'll just give you a couple anecdotes of what we've been doing in the lab. The first kind of concept, my first big NIH grant was go get every human disease studied by microarrays, okay? Just go get, and when I wrote this thing, I don't know, 10 years ago, it was like there was a whopping 25,000 of them freely available. Now we're like 2 million. And we started to collect disease and healthy, disease and healthy. You know, it's hard to find these kinds of experiments. A lot of people study, for example, metastatic cancer and non-metastatic cancer. We collected every experiment that also had healthy because we wanted to kind of line up all the healthies and compute a distance away from healthy. That's a disease. So pretend these are all genes, so these are amounts. I'm not going to show you any gene symbols. We calculate signatures. Everyone's been doing signatures for a long time. And the first thing we wanted to do, this is Marina's slide here. She's the best with graphics in my group. First thing we want to do with all these microarrays and these signatures is actually to compute or to calculate or discover diagnostics. So what's a good diagnostic? Well, a diagnostic tells you if someone has a disease. So a patient in front of you has a disease. There's imaging diagnostics. I love serum protein diagnostics. I love proteomics. But actually, I love 1970s proteomics, right? ELISAs. Remember those ELISA kits? I love ELISA kits because I was barely a pediatrician at one point, but it still amazed me that if I had a screaming child in front of me with a sore throat, somehow you could stick this Q-tip there and put some drops and you could tell if it's strep or not, right? I mean, or with serum proteins, for example, protein-based tests are just great because they can work in a desert, right? You don't need a cold chain or anything. So kind of simple non-radical thinking is if a lot of these chips are measuring RNA, if the RNA is different in a disease, maybe the protein is, and maybe we could detect some of that protein in the blood, circulating in the serum or plasma. That's the idea. So we did a bunch of these. We did some cancer ones. We did some organ transplant ones. I'm going to spend a second talking about this one, preeclampsia. Any of you have heard of this condition? Raise your hands. Yeah, okay, so it's, of course in this room, it's about half or so, let's say a third or half are raising their hands. Usually the only people who've heard of preeclampsia are the ones that watch Downton Abbey because one of the characters dies of eclampsia. Spoiler alert if you haven't seen this yet. Okay, but that's usually the only reason Americans know about this. This is when blood pressure goes really high in a woman who's going to have a baby, and you know this disease falls right between the cracks. The obstetricians, I love them, but they say just deliver the baby. The delivery is the cure, and the blood pressure comes down about 48 hours. And then we pediatricians got to keep these tiny babies alive for months. So it kind of falls right between the cracks. And we don't really even have a good diagnostic for this. We're looking at protein in the urine. It's kind of like what they've been using for 100 years. A lot of costs involved, a lot of hospitalizations involved. And we wanted to, forget about a prognostic, we didn't even have a specific diagnostic yet. So Linda Liu's a grad student who started with this work, and then Matt Cooper and Bruce Lane got involved. Bruce did the proteomics. And if you just search in the repositories for preterm birth or preeclampsia, 266 microarray experiments already run. Here are 299, 249, 154 samples, 143 samples. How many more do you need to get started here, right? I mean, look at all these samples, look at all these experiments, public, human. And the methodology here is really kind of simple, okay? I make everything seem easy, but the idea here is a massive Venn diagram. I'm going to put all these experiments together. I'm going to reanalyze all of them. And I don't trust any one of these experiments, okay? I love them, but I don't trust any one of them. But I trust what I get in common. Right? If 3, 4, 5, 6, 7 of them are seeing the same transcripts upregulated, then it's worth chasing them down to see if I can detect the protein for some of these. So I'm going to just put the best ones of these together. We can look at QC and stuff and chase down the ones that are in common. And you get markers like this. Here's an example, hemopexin. It's upregulated in women who are having preeclampsia compared to those that are not here. These are pregnant women who are not having preeclampsia. And that's one of about 7 or 10 different markers that we found. And these are just conventional serum ELISAs that you can buy. And we got a couple papers out. There's some links at the bottom there. What do you do next? In Silicon Valley, you start a company on this, okay? And you're going to see a couple of these arrows here. Start with the unmet need. You got the data available. We had March of Dimes money to get started. We analyzed and designed a diagnostic. We had one of these CTSA grants, Clinical Translational Science Awards. At Stanford, they were called Spark Awards, $50,000. We got some samples validated from UCSF, Stanford, some that we got off the internet. And then we launched a company, $2 million in seed financing. Now, let me be crystal clear here. I'm not showing you $2 million to brag about it. Actually, it's not a lot of money, okay? But $2 million, I'm not showing that to brag about. I'm showing you the $2 million here because I'm more convinced than ever before this is the new way science will continue out of academia. We've got our papers. We filed the IP. If we want to make a difference for patients, we're going to have to start a company and get this to a diagnostic. But the science is continuing in the startup company, right? We don't know if this is actually going to work prospectively in patients. Scientific experiment needs to get run, now funded with $2 million. So let me put it a different way. Do you know how hard it is to get a $2 million NIH grant today? The idea now is the science continues by private money, continues in the startup company. And that's the concept here, and that's why I don't think it's taboo to talk about these kinds of things. Now, one reason why I really start with this one, I love this one, is because we already sold this company. Some of you know what that means, right? So we went from public data to designing the diagnostic, testing the samples, launching a prospective trial with the $2 million, and sold the company in 24 months. Inventors happy, investors happy. Can we do more of these? Of course. And I'm giving away my secrets here because there's so much unmet need in medicine. Each one of us in this audience could pick a different disease and we would never step on each other's toes. And this is literally how we're doing it, right? We got the data. Can you come up with a diagnostic here and see if this actually works? We're kind of repurposing that data, not for the molecular biology side of it, but actually see if we can get a translational use of it. Now, the other next direction I'll talk about is the therapeutics side, which is also a really painful problem. If I've convinced you about the disease side, therapeutics are going to be really the unmet need in precision medicine. We really don't have enough therapeutics. In fact, we don't even have enough biotech and pharma companies in the world. Even if they're 100% successful, we have so many diseases in the book, and the book keeps getting larger, there are not enough companies in the world to develop the drugs we need. So by definition, that means we need a more efficient way to do this. And so if I've convinced you've got the disease samples, well, there were people starting to test drugs, like Justin Lamb, Todd Golub, and others at the Broad Institute, just testing cells with drug, without drug, with drug, without drug. So on the left, you got with disease, without disease. On the right, you got with drug, without drug. This is the connectivity map, links, a bunch of these types of projects. We're going to put these two together. So the reporters for this called this Match.com for drugs. If you know what Match.com is, it's how you find a date or a spouse. And the famous saying is opposites attract, right? So I'm going to show you all the bioinformatics with my arms. If I have a disease where this gene goes up and this gene goes down, and I can find a molecule on the right there that makes this one go down and this one go up, maybe there's a match there, right? That's 2 genes, imagine 20,000 genes. I'm not going to show Kolmogorov-Smirnov tests and stuff like that. And there are 2 C words I'm not mentioning here. One is we're not getting to the cause of the disease. I understand that, okay? I don't think we have a lot of drugs in medicine that actually treat the cause of the disease, but we're not getting to cause. And I'm not talking cure, okay, the other C word here. I'm looking naively for a treatment that might reverse, okay? That's the kind of idea. We had a bunch of these that we published. A bunch got us in the newspaper. My favorite is this one. This one came out a year and a half ago. Psychiatric drug, imipramine, okay? So this is not the concept of not just finding a new drug, but can we find a new use for an old drug? Imipramine is an antidepressant, and based on that approach, we computed it should work on small cell lung cancer, which has a 5% survival rate. Okay, 5% survival rate. Imipramine. So humans smoke to get small cell lung cancer. Or I guess you set up that Gatling gun we just saw earlier, right? But this mouse, you knock out P53, RB, P130, this mouse gets mouse lung cancer. So it's not a xenograft, it's not a PDX. This mouse it's a genetic engineered mouse model where this mouse gets its own lung cancer nodule. So this is Julian Alejandro. Joel did the informatics and Nadine did the work in the lab here. And this mouse is the same mouse treated with imipramine. Now, imipramine is an antidepressant. We don't even use it anymore. It's still in the pharmacy, but we use serotonin reuptake inhibitors like Prozac. We don't use imipramine because it's got side effects. It makes some people sleepy, and in others, it might set you up for what's called a QT prolongation. It might set you up for an arrhythmia. Actually, neither of those 2 side effects sound as bad as having lung cancer, 5% survival rate. And lung cancer is melted away. It's gone in this genetic engineered mouse model. Now, why am I showing you this one instead of the dozens we've done? Okay, I love this one. Because we start with public data, we make the computational predictions, we test the cell lines in a dish. I'm not going to show you that data. We get this amazing lung picture, right? 15 months after the computer prediction, we got IRB approval, we launched a clinical trial, and patients are dosed on this drug now. Total cost of that trial, $50,000. Okay? Not $1 million, not even $100 grand, right? That fits in one of these CTSA Seed Awards here, right? Now, it turns out this is a harder disease to treat, of course, than we've expected. Out of those two side effects, sleepiness is the worst of the two, it turns out, because if you're a patient that's told you only have 5 months to live, you actually don't want to spend it sleeping. I can understand that. You have a bucket list of things to do. So we got a lot of molecules we got to chase around there, but we got to do more of these. We cannot wait for someone to read our papers and then think about launching a clinical trial. We have to do this. We have to do this in a more efficient way. And the data is what I think is going to enable all of this to happen. And of course, you got to start a company on this in Silicon Valley. You have the unmet need, the data is available. First an NIH grant, company launched with ARA funding, and then StartX, which is a Stanford incubator, and then more than $5 million now raised. And actually, this company, Numeti, is now developing drugs for Allergan. Allergan, once they merge with Pfizer, will be the biggest pharmaceutical company in the world. And you got these teams of like 6 or 7 people developing the next psoriasis drug for the biggest pharmaceutical company in the world. So I use this term garage biotechs. I know we start Apple and HP in a garage. I think we could start the next Genentech or the next Amgen, maybe the next Merck in a garage, actually. And I don't mean making meth in your garage. I mean cancer drugs and diabetes drugs, right? So just to close things out, where do I see things going? If I had to tell you where to skate, you know, you got to skate to where the hockey puck is going, as the saying goes. What do I think is the next big open data? Clinical trials data. What's a clinical trial? The most expensive experiment in the world. They're usually well controlled. They're costing us billions, right, to develop these drugs. Half of them fail, and we have this weird culture that when they fail, we don't even write a paper about it, right? Never mind releasing the data. I think it's going to change, and I don't mean just putting that summary table on clinicaltrials.gov. I mean releasing the raw clinical trials data. EMEA, which is the European FDA, is starting to require this. I think our FDA will get there. And so I run this thing called IMPORT, which is for NIAID, allergy infectious disease. It's an allergy, we work with almost 15, 20 different consortia like Immune Tolerance Network and organ transplantation, a lot of vaccine studies. We release their data, but we release their clinical trials data as well. And so we're one of the only repositories actually in the world where you could just freely download dozens, hundreds of experiments, just raw clinical trials there. Imagine doing meta-analyses, meta-studies. This is the kind of work that we're thinking about, or even rescuing old drugs that have failed a clinical trial with this kind of data. So we're showcasing how to do that. Sign up here. You can download all the data you want, and this is growing like crazy. Now we have 500, 600 downloaders a month now. It's just amazing. And by the way, we have 60,000 flow files. I know you guys love flow. 60,000 annotated flow files. It might be the largest repository of flow cytometry. I'm not sure about that, but I think we're one of the largest here. All right, just to close it out, so some of you know I just moved from Stanford after 10 years to UCSF. I love Stanford. I love everything about Stanford. I have never had a bad day there. But the opportunity at UCSF is incredible. So I'm now the director of this Institute for Computational Health Sciences. We're slumming it out in this brand new building until they build our brand new building in 2019. Amazingly positioned between Illumina and the brand new hospital. What does the realtor say, right? Location, location. You couldn't even design a better spot for us than that. Between Illumina Hospital, some people think we should be a wall between these two. We're obviously going to be a conduit between these two. And that's fun. We get to recruit a dozen faculty, and we're building out like crazy. But actually, why did I move was because of something called UC Health. It turns out UCSF is one of 5 University of California medical schools. So it's UCSF, UCLA, there's San Diego, Irvine, and Davis. And all 5 actually have an organization called UC Health. In fact, people don't realize if one of them approves an IRB, actually the other 4 automatically approve it. It takes a couple of extra days, maybe a week or 2, but actually it's automatically approved. If one approves a contract, the other 4 already approve the terms. One of the amazing things about these is that I'm now in charge of putting the entire electronic data warehouse together of every single patient in the University of California system. If you do the math right now, this is actually a slightly old slide, we have 14.5 million patients that get some care in the University of California. That's about 4% of the US population. And now I get to build the electronic data warehouse for all this data. In fact, we're already done. I wanted this to be done before my first year anniversary, so we've actually finished this. That means all the Epic data, one of us uses Allscripts, and all that record data is all in one place. We can imagine a lot to do with this kind of data. We're imagining and enabling a lot of people. Clinical researchers to run a GWAS. Yeah, we know. We'll get to the genetics on the 14 million piece by piece, but we'll get there. Imagine a GWAS across the whole UC Health. Community activists could do data mashups with environmental factors or develop models of future disease. Cancer genomics researchers can figure out all the cancer cases we have in the entire University of California system. What I'm trying to build are maps, and I'm going to end with this concept. I love maps, and I'm trying to build maps of how Californians get disease and die. That sounds kind of morbid. Think about Google Maps, right? So here's an example of a map. This is from real data. We have patients that come in for alcohol-induced mental disorders. This is alcoholism. A year later, a lot of them end up with liver cirrhosis, and then some of them a year later end up with liver abscesses, or sometimes people go this way. And then the squares mean patients died of this. So this is work done by Hannah and Jay. Hannah's done the computation, Jay's done the graphics here. So you can see people die in different directions. Sometimes these maps are kind of complicated. Here, patients have a heart attack, a bunch of them die, a bunch end up with heart failure, that means the heart isn't pumping enough, and then they end up with lung diseases and sepsis. So I never really realized this, but a patient with a heart attack can die of sepsis, which is a blood infection, just 3 years later, okay? So you can see where people die along the way. So this is the idea of building maps. Now this is like Google Maps, but this is kind of morbid compared to Google Maps. Google Maps might take you to a pleasant destination. This tells you how you're gonna die. It's slightly different. But the problem with Google Maps in some ways is that Google Maps actually doesn't really show you every car on the road, right? It kind of shows you traffic, but it doesn't show you every car on the road. It kind of is still too much of an approximation. And my goal, when we open that new building in 2019, is we're gonna have a room full of monitors, right, on the screen. And we're gonna have our Google Maps of death and disease, and we're going to start to track all of our patients. And this is a prototype here. You can see as the age increases, how patients go from disease to disease to disease to death. Some of them are dying, the age goes up. This was real data in California. And we're going to track our 14 million patients. And we're going to predict what's going to happen in the next 90 days. And we're going to predict what's going to happen in the next year. And we're going to do something about it. And this, to me, is going to be the new definition of an accountable care organization, an ACO. One that actually accounts for the care of its 14 million patients. Not just when we see them in encounters, but all the time in between with mobile devices and wearables and all the rest. And this is just a prototype, but this is where we're going in the University of California. Can't do any of this work without a long list of collaborators here. Whoops, let's get the next slide here. 40 different people across the University of California helped me build all of this data together. I have a bunch of collaborators that have done all this work over the past 11 years, a bunch at Stanford, now increasing amounts at UCSF and all around the world, especially Alejandro Julian helped with that cancer mouse model. We're hiring like crazy. I got to put in a plug, associate full professors, so senior folks as well as postdocs. And I have to thank UCSF for endowment support. My lab is blessed with 17 NIH grants from these 9 institutes of NIH. These 4 give me more, these 5 give me less, but I still love them. March of Dimes, JDRF, HP, HHMI, stem cell money. We have disease-specific foundations that help us. I always thank my admin and tech staff. I would never get a grant or paper out the door without them. I always thank Zach for being my friend and my mentor for the rest of my life, Sam and Keith for recruiting me to UCSF. And I always thank my wife, Tharangini Deshpande. She's a molecular biologist, biochemist. She reads every major paper a grant comes out of my lab. She collaborates with us a lot, starts companies with me now, and lets me go all over the world to give talks like this. Thank you very much. Do I have a moment for questions? All right. Go for it.
Hello, just thanks. New England Journal of Medicine, research parasites.
Research parasites.
I figured that'll get a rise out of you.
Oh, it's already done that. Okay, so if people haven't realized, about a month ago, Jeff Drazen had this editorial, it kind of lasted for 48 hours, where he didn't call us research parasites because he knows people who call us research parasites, if you read it carefully. And so the concept is that, in fact, clinical researchers are going to be the last to get this, right? We in the molecular world have been sharing data for a long time. We reanalyze this data. We know. I mean, there's a culture here. The clinical researchers are the hardest. They've had things like Framingham Heart Study, Nurses Health Study, those kinds of cohorts, and they build their careers writing paper after paper. It's uncomfortable for them to release that to the public. Yet, actually, it's already released in dbGaP and some of these other kind of genetic repositories. So it's uncomfortable for them. The response to that was unbelievable, including the editor-in-chief of Science, who just had an editorial just 2 weeks ago. If you follow Twitter, you can look at the research parasite hashtag and just see all of this. But that editorial came on a Friday. Jeff had to kind of backtrack even that Monday with the roar against him. So look, there's this cultural thing that people want to protect their careers with the data, but it's changing fast. The world is just moving too fast for them. Other thoughts, comments? There's a question back there.
One of the interesting things about data is that it comes in quantity and it comes in quality.
Oh, yes.
In this area, how do you think about that trade-off?
Oh, that's usually the first question I get is how the quality of the data. Okay, so
well,
of course,
let's do both.
Yeah, so quantity is easy, right? So the data is, let's say, doubling every 2 to 3 years. And by the way, that means anytime you look at the repository, if the data is doubling every 3 years, that means by definition, half of what you see in the database today is 3 years old or younger, right? Because it's doubling every 3 years. So it's not like ancient data in there. Half of the data is always young, fresh data. So get that for quantity. Not only is each database growing exponentially, but the number of databases is growing exponentially, which is a double exponent, which is actually just an exponent. If you want to know where to find these repositories, there's the weirdest issue ever. It's called the Nucleic Acids Research. There's a database issue every January. And just to give you a sense of how many papers are out there, I think the last January issue had 1,600 papers in that issue of Nucleic Acids Research, just describing all the databases that are out there. And to put a size perspective, all of NIH's databases, all of NCBI were 2 of the 1,600 papers. So imagine 1,598 other major papers like this. So it's a massive amount of data. So that's quantity. Quality. What I realized and analyzing this kind of data is you have quality control. You can actually look at the quality here. But actually, I don't even worry about quality anymore. There are very few evil people in science, right? So most people generating the data are the best in the world, actually, right? In fact, University of Podunk doesn't get NIH grants, right? It's our institutions that get NIH grants. It's our peers. So they're usually the best scientists. But for some reason, the minute it hits the internet, we think it's valueless, right? Because we've been trained. If something is free and on the internet, it must be valueless, like these YouTube videos of kittens, right? I mean, that's the kind of culture we've been trained. This is the most valuable data in the world. Now, here's the last point I'll say. The problem is you could always criticize the data. But what I will say in response is that we're such good skeptics in science that I will easily bet you 50 years from now, we'll still find some way to criticize data collected 49 years from now, right? I mean, why would we say it's perfect at that point, right? So if we're always going to criticize the data, it's never perfect, right? Don't let perfection get in the enemy. It's the enemy of the good, right? Voltaire. But instead of picking on the data, my first papers in my career were all about, look at this microarray, doesn't match that microarray. Now the game is what can you do with the data in its current form? It's always getting more plentiful, and it's always getting better. But what can you do today? Don't wait for perfection. So it's my long kind of practiced answer for that one. Thank you very much.